So what I'm going to do is give you a little bit of history, as I often do, those of you who know me, and I'm going to tell you about the nucleic acids and the protein data bank. In the 70s, Rutgers, we used to call it DNA University. And in the 70s, there were many rising stars in the DNA universe at the time. Wilma Olson, who was studying DNA folding, Jerry Manning doing counter-ion condensation, Ken Breslauer doing DNA calorimetry, and Roger Jones DNA synthesis. And these folks ran meetings fairly regularly at Rutgers. And at the time I lived in Philadelphia, and I used to be really happy to come up to Rutgers to listen to what was new in nucleic acids. And at that time, Rutgers emerged as an intellectual hub for nucleic acid science. A little bit after that, Rutgers began recruiting structural biologists, and many studied nucleic acid containing structures. You just heard from Eddie, Richard Ebright. And when I came, I was working on DNA structure, Kathy Lawson on transcriptional regulation, Ann Stock, who you'll hear from, on bacterial response regulators. Millie Georgiadis was recruited as an assistant professor, worked on reverse transcriptase. Joe Marco Trigiano, who you'll also hear from. And then I guess it's been about 8 years ago, Stephen Burley, who was very well known for his work in the 90s on Tata binding proteins. As you also heard, lots of collaborations emerged from having these groups of people, the structural biologists, together with the people who were doing biophysics on nucleic acids. And so we began to build even more in the area of nucleic acid containing structures. Here is a little gallery of like a less than 2 handfuls of the sorts of structures that people were working on from within the Rutgers community, or have worked on from within the Rutgers community that contain nucleic acids. And some you've seen already, and it is a very sort of rich framework to work in if you were interested, as I was, in nucleic acids. So building on that, when I first arrived at Rutgers in 1989, and there were folks that I told you about from the beginning, Wilma and Roger and Ken, who were really into nucleic acids. One of the problems with the PDB back then was nucleic acids were not as well curated as the proteins. Not surprisingly, since it is the protein data bank, there were nucleic acids in it, but they kind of didn't get quite the treatment they needed. And so we got this idea to create a research resource for scientists studying nucleic acid structures. And the co-founders were myself, Wilma Olson, and David Beveridge, who is retired from Wesleyan, and actually was the mentor of a current PDB member named Rachel Kramer Green. So we're all a family, as you know. We had a scientific advisory committee. Stephen Burley was the chair for the NZB, and he told me, and I didn't listen, under no circumstances should you try to take over the PDB. So I didn't listen, and I'm glad. The accomplishments of the NDB is we established standards for nucleic acid geometry, because unlike proteins where there were well-established standards by Eng and Huber, nucleic acids were all over the place. We implemented a standard reference frame for the base pairs. The base pairs, as some of you may know, are not flat, and they have different kinds of conformations, and that's sequence dependent. And Wilma Olson and her group had studied this for many years, but there were other groups who were studying the same thing, and all the answers were not matching. And that turned out to be something relatively simple, but for some reason it took a trip to Japan and a workshop to figure out what was going on. And Jianjun Liu, who was working in Wilma's lab, came up with a standard reference frame that we adopted, and then we got all the standardization of these base pairs in order. We built a pipeline for processing nucleic acid structures that took into account all the features of nucleic acid structures, which, as we all know, are not the same as proteins. We created the first searchable relational database for biological macromolecules. Until the NDB, all you could do is get a bunch of flat files and kind of try to make sense of the whole thing. And we built the entire system using the emerging macromolecular crystallographic information file format, MMCIF. And as you heard from Stephen, John Westbrook was a graduate student in Ron Levy's lab at the time. And he got really interested in this subject and came up with the framework, which to this day is the one that we use. And it was really an amazingly brilliant set of insights that he had as to how to do that. And then we made ourselves an organization. We called ourselves the Research Collaboratory for Structural Bioinformatics, of which the NDB was the first member. And then when we finally did compete successfully for the PDB became the second member. I told you about the nucleic acid standards. The base sugar phosphate geometries were derived from the Cambridge crystallographic database, and those numbers are being revisited right now, but they're more or less correct. One of the problems is that not everybody was using them, and we have to fix that. We came up with standard coordinate sets for monomer units so people could do modeling of nucleic acids. And then a big innovation was new parameters for the refinement of nucleic acid containing structures, which were very difficult to refine. As soon as you had a protein and a nucleic acid, the refinements did not behave properly. And Gary Parkinson, who was in my lab at the time, worked together with a group of people to come up with these force constants. And we were tutored, mentored, and told what to do by Axel Brunger, one of the great people in structural refinement. So that then became something that people began to use, and the refinements started to behave properly. The standard base pair frame is shown here. This is the Jianjun's drawing that's in the white paper that we produced. And you'll see some familiar names of who was at that meeting. And Stephen was there. I was there. Eric Westhoff, Cynthia Wahlberger, and Wilma. Wilma was the kind of instigator of the whole thing because of all this problem with having the non-standard base pair frame. So we built a pipeline for data processing in the nucleic acid database. In principle, we became a primary archive for nucleic acid structures, and then we handed the structures over to the PDB, which at the time was at Brookhaven. In fact, that's not really what happened, but it doesn't matter. We built this pipeline. MMCIF was the underlying format. It was generalizable for all macromolecules. And when we finally did get the contract for running the PDB, one of the first sets of structures that we got were the ribosome structures. And we were really uniquely qualified to be able to process those structures, which contain a huge amount of RNA, because we had been curating all this data for the NDB. So what did the NDB provide? It provided a lot of torsion angle analysis, base morphologies, something called RNA view, which was done by a former member of the PDB named Juan Wang, and you could then get a much better sort of two-dimensional picture of where the base pairs were in the real structures. We curated structural and functional classes of the structures in the NDB. We did a lot of work on structure similarity, created a lot of tools and software. And then we had link outs for conformational analysis to DNA TCO, which is now run by Bodan Schneider, who at the time was working back in the old days. He was a postdoc of mine, now is the director of an institute in Prague. And then when I retired and moved to California, I worked on a program with a student in Remo Ross's lab to really get much better description of protein nucleic acid interactions in these structures. In 1999, RCSB assumed the management of the PDB, and the RCSB PDB used the data processing infrastructure created by the NDB. The NDB then became a knowledge base with links to the PDB. So the PDB was the primary archive for all biological macromolecules, and NDB became a knowledge base with various features that you could search for. So right now, the PDB has a significant diversity of nucleic acid structures that are properly processed thanks to the very early work that was done on nucleic acids. So I'm just going to go through the classes, types of structures that exist. There's about 2,100 structures with DNA alone. The early days were a lot of small helical fragments. Quadruplexes became increasingly important, and a significant amount of that work was done by a colleague and collaborator of ours named Stephen Needle. Holiday junctions began to appear, and a fellow graduate student of mine back in the old days in Pittsburgh, Ned Seaman, designed these three-dimensional crystals, and they're just beautiful structures, and was basically the father of this nanocrystallography, and made important contributions. And very recently, there are these DNA zymes, which are like ribozymes, except they're made of DNA. And so that is now in the PDB. For the DNA protein, there are, so I should say the number of DNA structures in the PDB is growing at a very modest rate. For the DNA protein, they're growing a little bit more. There are 5,700 structures. We've classified them as to whether they're regulatory enzyme or structural, and of course, viruses. And these are some examples. For example, P53 DNA complex. There are many of those, and a very important set of structures. DNA polymerase, and then, as Stephen showed you, the nucleosome. And so these are examples of the types of DNA structures in the PDB. RNA alone, as you know, back in the old days, nobody thought RNA did very much, and it turns out that RNA is has very diverse functions. There are about 1,500 structures. They're mostly single-stranded. There are some small fragments that are double-stranded. Interestingly, tRNA was one of the very earliest structures that went into the PDB in the 1970s, and is, of course, a key structure. And we've learned over the years that the RNA world is multifunctional, providing both information, and it has the ability to be catalytic. And that's all over the last many years we've learned that. And by far the fastest growing group of structures in the PDB that contain nucleic acids are RNA protein. Right now, there are about 2,600. It's the fastest growing class of nucleic acid containing structures. They're very diverse, and they have many different functions. And of course, we have the ribosomes and viruses and enzyme complexes and many, many more. And they're just really beautiful structures. This picture, every year, Kathy Lawson is asked by the RNA Journal to produce cover pictures of these structures, and this is an example of one of the very beautiful structures that she drew. So the new developments in the nucleic acid world are the Bodan Schneider, who started out with us and is now leading up a group with Elixir in Europe, and new valence geometry standards are being created by this working group to get everything realigned because they had really gone off the rails. There's new conformational validation being done using virtual bonds. Brenda Vellet and Kathy Lawson have done that. We have new functional annotations. As I told you, the core archive is the PDB. And then the NDB had been funded in the early days and then basically was in maintenance mode. A couple of years ago, we got funding to revitalize it, and we've turned it into something called the Nucleic Acid Knowledge Base, which will contain these value-added annotations and will be linked directly to the PDB. So when you get a nucleic acid-containing structure, all of these extra annotations will come along with it. So here's this example of a new validation scheme, which Brenda and Kathy are working on, where you can see if basically they are trying to make what is the equivalent of a nucleic acid Ramachandran plot using these virtual bonds. And this we can trace all the way back to Wilma, who was kind of the pioneer in virtual bonds in nucleic acids. And this is just a sketch of what the NAKB will look like. And it'll be very much more easy to navigate than the current NDB, and will allow you to have this extra information. So acknowledgements, I need to and want to acknowledge Wilma and David, who worked with me in those very early days to establish the NDB. And then there's Bodan shown here when he was working for the PDB in the 1990s. And of course, John was the really the brilliant architect of MMSIF, and he was extremely important in the development of the NDB and figuring out how to do this the right way. Anka Gelbin was our kind of chief and only annotator. Christine was our outreach coordinator. Sushin Shea, Thomas Demini, Shri Jane, Zukang, Feng, and Li Chen. Some of the people are still working on the NDB. And the emerging NAKBs, Kathy, Craig Zerbel, who is at Bowling Green, and unfortunately, Neocles Leontis is no longer with us. He passed away last December. And then the whole RCSB PDB team has always been working they've, as I said, working on both proteins and nucleic acids. The funding, the early funding for the NDB was all from the NSF in various ways, and more recently, the NIH. And with that, I will stop and thank you for your attention.